All right, so we have 3 lectures dedicated to the topic of blood. And in this first lecture here, we're going to talk about red blood cells. And just to give you an overview, so blood is a specialized connective tissue, and it has a total volume in the adult human of about 5 to 6 liters. The fluids in the body, they're separated into 3 compartments. So you have the intravascular, which is actually in the blood vessels like this. And then you have intracellular, which would be like this, in the cell like this. And then what you have is interstitial, which is in here, which would be this space between the blood vessel and the cell. So this is interstitial fluid. And then just to give you a breakdown, so these together would be considered extracellular fluid, or ECF. And so the intracellular fluid, or ICF, is about 2/3 of the fluid, or the fluid volume within the entire human body. And then the ECF is the remaining 1/3. So the majority of the fluid in the body is actually within the cells. So peripheral blood, when it gets centrifuged, it gets separated out into 3 layers. So the first layer is the red blood cell layer. And so it would be down the most, remember when you centrifuge something, the most dense mass is going to be found at the bottom. And so this layer here is going to be all the red blood cells at the bottom. And this is going to be about 45%. So a little less than half. And then you're going to have this kind of whitish layer, which will be your white blood cells. This will be about 1%. And then the rest of this is going to be all plasma, which is the fluid component, and that's going to be about 55%, the remaining percentage. So plasma, it's the liquid component of blood. It's composed of almost entirely water, greater than 90%. It also contains plasma proteins such as albumin, fibrinogen, immunoglobulins, or antibodies, prothrombin, clotting factors for the clotting cascade, electrolytes like sodium and potassium, lipids, amino acids, vitamins, and carbohydrates. And then erythrocytes, so these are measured clinically in terms of hematocrit, which is the percentage of red blood cells in the blood, and then hemoglobin concentration, which is measured in grams per deciliter. Something to keep in mind for both of these, the range, the normal range is going to be a little bit higher in males than in females. So in males, it's 40 to 50% is your normal hematocrit, and then 30 to 45% in females. Normal hemoglobin concentration is 14 to 18 grams per deciliter in males, and then 12 to 16 grams per deciliter in females. The other thing is mature, so red blood cells are produced in the bone marrow through a process called erythropoiesis. And so when you get to the mature stage, when the red blood cell has actually exited the bone marrow, they have a lifespan in the circulation of about 120 days. Erythrocytes are also 7 to 8 micrometers in diameter. And as a result of that, because they have a consistent diameter, they're often used as what's called a histological ruler. So if you look down here, here's a peripheral blood smear. It's full of red blood cells. So if you look at here, this would be about 7 to 8 micrometers. You compare it here to this lymphocyte, so you can kind of get an idea of how large this lymphocyte is. So mature erythrocytes, they're anucleate, meaning they don't have a nucleus, and acidophilic. That's what gives them that eosinophilic appearance, that reddish pinkish appearance. They also do not contain many of the organelles you see in other cells. So they don't have ribosomes, they don't have endoplasmic reticulum, they don't have a Golgi apparatus, and they don't have mitochondria. They have a biconcave shape, which would look something like this. Versus a sphere. There are some diseases called spherocytosis where you can have spherical shaped red blood cells, but that is not normal. So they have a biconcave shape, and this allows for maximum surface area to cytoplasmic volume ratio. This enables for 2 things. One, so that they have efficient carrying of the gases, oxygen and carbon dioxide, for gas exchange. And then also it allows the membrane to be flexible along with the cytoskeletal component, which we'll talk about in a second here. It allows it to be flexible and elastic to allow red blood cells to bend and contort, allowing easy movement through capillaries. Because you got to remember, you go from large blood vessels to medium-sized blood vessels, small blood vessels, and then you go to capillaries, which are very, very thin, about wide enough for one red blood cell to pass through at a time. And so if these can bend and kind of squeeze through these very easily, it keeps them from getting caught up in here and blocking blood flow. Like we kind of mentioned, the main function is to carry hemoglobin, which transports oxygen and carbon dioxide to and from tissues respectively. And then also, erythrocytes exclusively carry out anaerobic respiration because they don't have mitochondria. You need mitochondria to carry out aerobic respiration. So as a result of that, erythrocytes, they rely on glycolysis exclusively for ATP production. The other thing about red blood cells is they contain a network of cytoskeletal and transmembrane proteins that maintain this biconcave shape and also enable the flexibility. So if we look at the membrane like this, you're going to have these transmembrane proteins spanning the entire membrane, and then you're going to have this whole cytoskeletal network. And then if you were to look at the undersurface of a membrane, let's just say like this, you would see that, the transmembrane proteins, this is if, they're coming out of the page at you. And then you would see they kind of have this hexagonal shape like this. And they form this very complex network like this. And this network, it forms what's called this two-dimensional hexagonal lattice network on the inner layer. And it includes proteins such as ankyrin, spectrin, actin, adenosine, and tropomyosin. And then these proteins form this inner cytoskeletal network. And then they also, some of them help connect to these transmembrane proteins such as glycophorin C and BAN3. BAN3 specifically has a couple functions. It's an anion antiporter, which means it exchanges one ion for the other, and specifically negatively charged ions or anions. So it exchanges bicarbonate for chloride. It also binds to hemoglobin. And then again, like we said, it provides additional attachment site for the red blood cell cytoskeleton. The ABO blood group system. So this is what helps us distinguish different blood types. So if someone's type A or type B, it helps guide transfusions clinically. We'll explain specifically why that is in a second here. So first, what it is, the ABO blood group system, it's essentially each of these is a glycoprotein. So these are 3 different glycoprotein antigens. An antigen is basically just a protein or a protein component that's found throughout the body, and it's also found it comes into play in immunology, and we'll talk more about this when we talk about lymphocytes later in the lecture. You can have foreign antigens, which are components of bacteria or viruses, and basically with every antigen, you can form an antibody. So the antibody will essentially tag it, and the thing is, if you get tagged, the reason why this is good for foreign antigens is it's how we fight infections. So you have an antigen from a bacteria, you tag it with an antibody, that antibody can help guide the immune system to clear this infection. Now, as far as native antigens, you typically do not produce antibodies to that, because if it's native to the body, there's no reason for the immune system to clear it. And so depending on the individual, they may have one or some combination of all of these glycoprotein antigens. As such, if they have the antigen native to them, they will not have antibodies. You'll see what I mean by this in a second. So these glycoproteins, they're found on the surface of the red blood cell membrane. And like I said, they're found in 4 combinations to form the 4 primary blood groups: A, type B, type AB, and type O. And these glycoproteins, they're attached to the transmembrane glycophorins. So if we come down here, here's the red blood cell membrane shown here, the bilayer. So here's the O antigen. And here's the glycophorin, which is a transmembrane protein, which we talked about on the other slide. And then here you have this sequence of sugar molecules, essentially. You have glucose, galactose, N-acetylglucosamine, fructose. And so all humans have the enzymes that'll catalyze the synthesis of the O antigen. So this is the O antigen right here, this sequence right here. So everybody produces this, everybody has this. So as a result of this, since this is a native antigen to everybody, no one's going to have antibodies. In theory, no one's going to have antibodies against the O antigen. So no one's going to have antibodies, so no one's going to have antibodies that would target the immune system to attack this specific antigen. Now, individuals with the A antigen, so everybody has the O, but if they have an A antigen, if they're type A, so type A blood, they're going to have an additional enzyme called N-acetylgalactosamine transferase, which will catalyze the addition of N-acetylgalactosamine to the O antigen. And so that'll yield, so this sequence right here is the same as here, but adding this on, that'll enable you to have the A antigen. And so these will be type A blood. As a result of that, though, someone who has type A blood is going to have anti-B antibodies. So over here we have the B antigen. So if someone's type A, they're going to have antibodies that'll target B. And so as a result of that, they cannot receive blood from type B. There's also type AB, which we'll talk about in a second. So they can't, because AB has both. They have A and B antigen, so they can't receive from them either. Individuals with B antigens, they possess a different enzyme, galactose transferase, which is an enzyme that will add a galactose onto the O antigen, and that yields the B antigen. And so as a result of that, these individuals will have anti-A antibodies. So as a result of that, they cannot receive from type A or type AB, because type AB is going to have both A and B antigen. And we'll summarize this in a second on here, who can give to who in a minute. So individuals with O blood type, they actually lack both the enzyme N-acetylgalactosamine transferase and the enzyme galactose transferase. So they can't make the A or the B antigen. However, as a result of that, since they only have O, they actually possess both anti-A and anti-B antibodies. So individuals with type O are what's called the universal donor. So they can give to A, they can give to B, and they can give to AB, and then obviously they can give to other people who are type O. They cannot receive blood from A, B, or AB because they have antibodies to A, so they can't receive from type A or type AB, and then they have antibodies to B, so they can't receive from B, and then again from type AB as well. Lastly here, individuals with AB blood, so they have both the A antigen and the B antigen. They have both of these enzymes, N-acetylgalactosamine transferase and galactose transferase. So they're going to have all three antigens, and then they do not have antibodies against A or B, and so they're the universal acceptor. So they can receive blood from A, B, other people with A, B, and then people with type O. And then as far as who they can give to, they can only donate blood to other people with AB. Because if they give to A is going to have the antibodies to B. If they give to someone with type B, they're going to have antibodies to type A. It's just not going to work out. So if we go to the whiteboard here and we just summarize this, so we have type, so we'll say can donate and can receive, just to keep it really simple here. So you have type O, which can donate to everybody. So they can donate to A, B, AB, and O. They can only receive from type O. So then you have type A. So they can donate to type A and type AB. And then they can receive from A and O. They can't receive from AB because they have antibodies to B. Then you have type B. So they can donate to B and AB. Because remember, AB doesn't have antibodies to A or B. And then they can receive from, obviously, B and O. And then lastly here, AB can only donate to other people with AB, because if they try to give it to A, problem is A has antibodies to B. If they try to give it to B, the problem is B is going to have antibodies to A, so it's just not going to work out. And then they can receive from, however, they can receive from everybody. So they can receive A, they can receive B, they can receive AB, and obviously type O. So this is important clinically because you don't want to transfuse someone with the wrong type of blood. because it can create an immune reaction and can kill somebody potentially too. And so again, this is used all the time in the hospital. You're going to always constantly, if you're going to be transfusing a patient, you always have to determine their blood type before you're going to transfuse them. Now, as far as the histological appearance of red blood cells, they're going to be round, eosinophilic, and anucleate. They're also going to have what's called central pallor, which is basically here, we'll show you down here. So you have a red blood cell like this. You can see it's kind of roundish shape. Obviously, they're very pink, eosinophilic. There's no nucleus because obviously in other cells you use the nucleus to kind of help you determine. You can't do that with erythrocytes because they don't have a nucleus. The other thing is you'll notice you have here and here. This is what's called central pallor, where it's kind of more whitish or less intense eosinophilic, or less, red or less pinkish here in the center. And that's actually a reflection of the biconcave shape. So you have the biconcave shape like this. And that central pallor actually corresponds to this thinner region in the center here. And notice how it's in the center. And so that's what you see here. You also can kind of see it. So this is a peripheral blood smear. This is actually a section that includes a blood vessel. So you can see the muscular layer here of the blood vessel, and here's the endothelial layer here. And so if you look at these blood vessels, if you can kind of see that rim shape like that. You can kind of see the shape like that and here. And so this is another way you can see, and that's another good example here. Here's that central pallor and then that kind of darker rim on the outside. And so you can see that here throughout. Again, here's another good example right here, where you have darker on this outer rim and then more central pallor in the center here. And again, that's corresponding to what you see on this darker outer rim is these two sections here. And then that center portion is this more narrow portion here. And so this is another good view of it here in an actual blood vessel from a section. So reticulocytes, these are actually immature erythrocytes. So you have, the bone marrow like this. This is a crude drawing of a bone like this. Let's say, this is a long bone like this. And so you have the bone marrow contained within here. And this is where you have erythropoiesis, and so from here, when you get release, you have initially reticulocytes. And then eventually, these mature into red blood cells, or also known as erythrocytes. So these reticulocytes, after they get released from the bone marrow, they circulate in the blood for about 1 day before maturing into red blood cells. So this is for about 1 day. And then eventually they get turned into mature red blood cells. And histologically, the appearance of reticulocytes, they're going to contain a blue hue to the cytoplasm. So they're not going to be as brightly eosinophilic or brightly red. And this is due to the fact that as red blood cells go through erythropoiesis in the bone marrow, and even at the stage of a reticulocyte, they have some ribonucleoprotein material. And as they mature, they continue to shed that away. And even at the reticulocyte count, they still have some residual ribonucleoprotein complexes. So they still have some nucleic material. And as a result of that, that's going to stain more bluish. And so that's what's going to give you that bluish hue to the cytoplasm. And so that's how you can histologically differentiate it from a mature red blood cell. And then eventually these remaining complexes get completely shedded out. And then that's where you have your mature red blood cell. Clinically, what you have is what's called a reticulocyte count, which is where you measure the amount of reticulocytes in the blood. And since it's the initial form of a red blood cell, when it's very first released from the bone marrow during production, it's actually used as an indicator of bone marrow function. So you have a normal level of reticulocytes, that's going to indicate that you have a normal level and a normal amount of production of red blood cell or of erythropoiesis going on within the bone marrow. And so this is really important when you're evaluating patients with anemia. So the normal reticulocyte count is going to be about 0.5% to 2.5%. If you have a lower count, this can indicate decreased red blood cell production within the bone marrow. You can see this in a number of different conditions. You can see it in patients who are undergoing chemotherapy undergoing radiation therapy, because those actually significantly damage the bone marrow, because remember from cytology, they damage areas where there's high levels of cell division. The bone marrow is certainly a place where it's constantly producing white blood cells and red blood cells. So it's an area where there's constant cell division, and so it's going to definitely get hit by cytotoxic chemotherapy or radiation therapy. So unfortunately, patients undergoing cancer treatment often have low reticulocyte counts and often are anemic because of that reason. And from the standpoint of if a patient is found to be anemic, which is essentially just a low red blood cell count clinically, if they have a low reticulocyte count, it tells you that it could be an issue with the bone marrow itself. Versus if they have a normal reticulocyte count and they're anemic, it tells you that the anemia could be due to something else. It tells you that the bone marrow is normally producing red blood cells, and the reason why they're anemic is for some other reason. It could be from decreased iron. That's a very frequent cause of anemia. It could be from a GI bleed, so like a peptic ulcer in the stomach. It could be slowly bleeding away, and as you slowly lose blood within the GI tract, that can cause you to become anemic as well. And as you can see, these are, external to the bone marrow. These aren't reflection of the bone marrow function. The bone marrow can produce normal amounts of red blood cells, but if you have decreased iron, or if you have, you're slowly bleeding out over time in the GI tract, that's going to cause you to become anemic. However, if it's elevated, which is called a reticulocytosis, this can be seen with increased red blood cell production. Now, there's actually a molecule called erythropoietin, or EPO, and this actually stimulates RBC production, or erythropoiesis. And what's interesting is there's tumors that secrete this. There's certain kidney tumors and other types of vascular tumors that will secrete EPO, and you'll see increased red blood cell production, and you can see an elevated reticulocyte count as well. Now, you don't want to have too many red blood cells, because you have too many red blood cells, that can actually clog up capillaries and arteries and lead to other problems as well. And so, again, the main thing with reticulocyte count is it's just used. What the bottom line here is, you want to know that it's a marker of bone marrow function. That's the main takeaway. Because if someone's anemic, you want to know, is it due to the fact that they're not producing enough red blood cells, or is it due to some kind of external factor? All right, so that concludes our discussion of red blood cells. In the third lecture for blood, we're going to talk about some clinical pearls relevant to red blood cells, so anemia, sickle cell disease, and how to read a CBC. But before we do that, we're going to cover in the next lecture white blood cells. Thank you for watching this sample video from our histology video course. You can access the entire video course, along with the histology lab videos, practice questions, and outline format ebook on our website, dviacademy.com, which is linked in the description below.